COVID-19 has shown the limits of the test methods used so far. They are neither applicable at airports and railway stations, nor can they be evaluated quickly. Moreover, they require a lot of resources, personnel, and specialized clinics. For Sandrine Gerber at EPFL, Igor Stefanini and Francesco Bertoni and their research teams, it was therefore clear that a faster and less resource-intensive testing method would represent a breakthrough in controlling the spread of COVID-19. In their project, the researchers worked on a device that could overcome the limitations of the test methods used to date.
So we developed a portable microfluidic device which screens for SARS-CoV-2 virus from saliva sample. It's using only very few resources. You do not need specialized personnel to run it, and then it can be deployed in, for example, developing countries where you cannot have access to scientific environment.
This microfluid device is a realistic prototype of a mobile, easy-to-use, and all-in-one laboratory instrument and has been developed over the last 2 years.
Faster screening of travelers helps to identify and react to potential pathogens at an early stage. This means that the spread of disease can be contained much more quickly than with time-consuming PCR tests.
The new device not only makes the process faster, but also much easier, as large quantities of tests can be processed simultaneously with the microfluid platform.
With the biosensor, we can detect the presence of the coronavirus RNA directly inside a saliva sample, and this can be done in a reliable and sensitive way. And this is easy to do because we put some probe fixed inside the device, and this probe recognizes the RNA of the coronavirus. So when the virus binds the probe, it gives a signal, and we can measure the signal via fluorescence or bioimpedance. So if there is the virus, we get a signal and we can measure it.
The advantages that the newly developed device brings also convinced the Swiss Federal Office of Public Health, which has supported the project with more than 1 million Swiss francs.
So up to now, we developed this device directly for SARS-CoV-2 virus, but as you can just change the probe and then you have access to other type of pathogen. Especially, we would like to develop the system to test several pathogens from the same sample from a disease which would have the same symptomology. And then we envision the implementation of this device in areas where you would have a lot of traffic, such as airport, train station, and ports.